Hello and welcome to Erik Penser Bank. My name is Claes Paulin and I'm an analyst at the bank covering the healthcare sector, and today's company, which we are hosting a company presentation for, it's Spago Nanomedical, and to present the company we have the company CEO Mats Hansen. Very welcome, Mats, and the floor is yours.
Thank you very much, Claes, and a warm welcome to this presentation, which aims to be a comprehensive company update and project update following on the release of our first quarterly report for this year. So very briefly, Spago Nanomedical is a Swedish nanomedicine company. We're now in clinical development phase. We have a technology platform that allows diagnostics and therapeutics of life-threatening diseases. And we are listed at Nasdaq First North here in Stockholm, and our aim is to become a leading nanomedicines company for precision medicine in diagnostics and therapeutics. So we believe we have all the elements in place to achieve this in a good way, and we will go through all of these items during this session. The technology, we have the evidence that the technology works. We have a good pipeline of projects that are now rapidly progressing. We can see a large medical need and a good market for these projects. We have a very agile and good team in place with high competence in these areas. And we have a position with strong ownership and a place in the market that suits us well. So with that, the agenda for today, we will start by an introduction to the company. I already started that. We'll go through some of the highlights from the report that was released today. We will also get an overview of the platform and the projects. Then we'll dive more deeply into the projects Tumor and Spargopix. And here I will hand over to our Chief Development Officer, Paul Hargreaves, who will speak to the projects on the scientific part and the clinical planning part. And we'll also go a bit into depth into the market potential and business development of the projects. After that, we'll summarize and we'll take some questions at the end. So with that, let's start with the highlights from the report that was issued today. We had a very good start of the year, I would say. We were able to communicate positive results both in our Spargopix project, but also in our Tumor projects. So what we have shown with Spargopix, and we'll obviously come more into detail on this later on, is that we have a very good safety profile so far in the breast cancer patients that we are currently looking into. And we've also shown that we have a very good contrast enhancement without background enhancement in breast cancer patients. So this is important not only for the project itself, but also to validate our technology platform. We'll come more into that as we go along. With Tumor, we were also able to communicate very positive data. We have shown that Tumor has the ability to slow down and to prolong survival in a preclinical model of colorectal cancer. This builds upon previous results that we communicated last year that we've saw good tumor controlling effects in a breast cancer model as well. So now we have two different models that shows the same type of results with Tumor, which is very encouraging going forward. We have a relatively good cash position with around 40 million at hand at the moment. And we aim to use this in a way that we promised the market a year ago to complete the Spargopix trial and to take Tumor to the start of clinical trials. So with that, let's go into the technology platform and what we have. I think first of all we should say that Spago Nanomedical is built around the convergence of nanochemistry, radiology, and nuclear medicine, which is quite unique for a small company like us. We have taken these elements or taken the fundamentals of these different disciplines and built them into one technology platform. And this technology platform is based on a biological principle called the EPR effect, the enhanced permeability and retention effect, which means that particles of a certain size accumulate in solid tumors by means of physiological leakage into the tumor tissue. That has allowed us to design very precisely nanoparticles that circulate in the body and accumulate in tumors in different places in the body. And this is a general phenomenon among solid tumors, which means that we are able to target very precisely different types of tumors, be it breast cancer or colorectal cancer or you name it. So it's a general platform that allows us to target solid tumors throughout the body. And how does this work, or does it work? Well, it does, and we've shown very consistently among the breast cancer patients that are in the current clinical trial that is ongoing, that we get very good and selective accumulation of our particles in the breast tumors. And this is not a given looking at historical data where this type of EPR effect has been claimed to be heterogeneous. So we're very happy to say that we have had good accumulation of particles in all the patients we have tested. And this goes very well along with the results we have seen in animal experiments as well. So that means that by showing this in Spargopix and by having good preclinical data with TumorAd, we have now opened up for our technology platform for use both in diagnostics and therapeutics of solid tumors in humans. So based on this technology platform, we are running two projects, and I've already mentioned them here. We have Spargopix, which is a tumor-selective contrast agent for MRI. And we have TumorAd, our upcoming radionuclide therapy project, which is now rapidly approaching clinical trials and will position us as a truly clinical development company, as we will have two projects in clinical development phase. And we'll now come in more in detail to each of these projects. And we'll actually start today by looking at TumorAd. And I will hand over the presentation to our Chief Development Officer, Paul Hargreaves. Welcome to take it away, Paul.
Thank you, Mats, and good afternoon to everybody. So this slide is really showing the basis for the TumorAd, where we have a cold carrier where we add a radioisotope of lutetium-177 to make a radiopharmaceutical. Next slide, please. So the actual clinical concept can be summed up in 5 basic steps. Firstly, we have developed a nanoparticle that's optimized for the physiological targeting of solid tumors. We then have selected something that we know is clinically effective as a radioisotope with the lutetium. The actual preparation of these functional nanoparticles is then very easily done either at the hospital site or at a central site. We then look to have the accumulation of the radiopharmaceutical in aggressive tumors and metastases, which is what we'll be testing in the clinical trials. And we know that irradiation of a tumor will reduce the risk of recurrence. Next slide, please. So our differentiation points of what's in the market at the moment are many types. We have a selectivity for malignant lesions due to the physiological tumor targeting that we find with this type of molecule. The actual treatment concept is proven. We're using a clinically effective isotope that has no specific need for drug delivery other than the cold carrier. We've optimized the treatment by the patient selection. So by using companion imaging diagnostics, we can see which patients are likely to have the best effect with the nanoparticle. We're also suitable for using combination therapy. Currently, radiation is used in combination with many chemotherapy targets. For us, we would be able to be part of those clinical regimes going forwards, as well as being a mode on its own. And we have facilitated logistics because our cold carrier, the nanoparticle, is non-radioactive. So that kit can easily be labeled either, as I say, centrally or at the local hospital. Next slide, please. So these are some more pictures that you've seen. We've got physiologically the radionuclide we've seen in animal models that it actually collects within the solid tumor. We've got excellent uptake in these tumors models as we've seen. We've also been able to see that not only is the uptake of the molecule in the solid tumor, but we're also able to actually differentiate between the survival of the animals that have been treated or not treated favorably for us with Tumorad. And as we've said already, the actual clinical results that we saw in Spargopix confirm that we have tumor accumulation via the EPR effect for this type of nanomaterial platform. Next slide, please. So here we have a regulatory toxicity studies have now all been completed, the so-called CTA required preclinical studies. We've seen the Tumorad tested at multiple dose levels. We've been able to incorporate many safety pharmacological endpoints and have seen that we have a very good safety profile. And we've been able to find the no adverse event level with a very large margin to those that we plan to take forwards into the actual clinical trial. We've also done some dosimetry experiments, which allows us to understand before we go into humans what the expected doses are for both the tumor and into other organs in the body. We've very much based this on what we've seen around the biodistribution of the molecule and its excretion within the rat model. And we've been able to see where we believe we need to be most careful with what are considered to be the dose-limiting organs. So this makes sure that we don't overdose the patient in areas which would then cause them multiple problems on top of their cancer. And then the efficacy, we've, as I've said, seen anti-tumor efficacy. We've seen the accumulation through the technology of SPECT imaging. And we've already proven positive results in 2 animal models, that of breast cancer and also that of colorectal adenocarcinomas. So we feel that with the CTA enabling studies, we're at a very good point to move forwards into man now. Next slide, please. So, as Matt said, we're executing on the plan through into clinical development. We have now the preclinical data. We've got the Spargopix clinical results, which gives us more supportive data around the EPR effect. We're also continuing to expand our preclinical efficacy program, looking at other tumor models for other tumor types. We are currently at a very advanced stage around the preparation of the clinical trial for patients. And these patients will be very much in the late stage cancer area. The IND for America and the CTA for Europe, enabling preclinical toxicity, safety, and additionally dosimetry programs have all now successfully completed. And we're compiling the regulatory documentation that will allow us to actually move forward into that first clinical trial. And we still believe we're very much on target to start the phase 1/2 protocol within this year. Next slide, please. So a little on the actual clinical trial. We're taking this as a two-stage protocol. So the first stage is a dose escalation where we will try the radiopharmaceutical at a number of different dose levels, starting very low and moving up to see where we can actually get the highest dose possible to give the most chance of efficacy in the patient. And then for the part 2A, or the phase 2A as it's known, we will go into specific tumor types. Here we will select the tumor types based on the aggressiveness of them, the market need, and also balancing the difficulty in actually recruiting some of these tumor types. We have decided that the study will initially be conducted in sites across Scandinavia, so we're keeping relatively local with that because we feel that's an excellent way to understand even more about the potential of the radiopharmaceutical. We've defined the patient population. So for phase 1, we'll look to around about 30 patients who have progressive disease. So this means that they have no more options for registered therapies. And then for the phase 2A, again, it will be the similar sort of late stage patients, but as I say, these will be in specific tumor types. Next slide, please. So this is a slightly busy slide. I only look at the middle column of this, but basically for the phase 1 part, which will take around about 18 months, we're looking to very much study the safety and tolerability of the molecule. Here, the actual outcome from that protocol or that stage will be that we will have a dose that we can take forward to in the actual real treatment stages. And then in the phase 2A part of the protocol, we will continue to understand the safety and how tolerable the molecule is, but we will start to assess the actual preliminary efficacy of Tumorad as a single agent in these different cancer types. Next slide, please. So the concept of Tumorad is very much that we need to have a long circulation time. So we have a molecule that does not get excreted particularly quickly. This allows the actual EPR effect to take effect, that we can actually get the accumulation that we require. And we find that we have maximum tumor accumulation in the non-clinical animals at between 24 and 168 hours post-dose. And importantly, this is a plateau. So unusually in this type of drug, we're going to keep a very constant level from the single injection. And as Matt said at the beginning, we have the option to actually target multiple tumor types as we move forwards. And I'll hand back now to you, Matt.
Thank you very much, Paul. And Paul will be back later on as well during this presentation. I will follow on what Paul said last here, that with Tumorad, we have the ability to target multiple indications. And we've looked very much into this, and we've had advice from medical advisors, and we've looked in the literature and so on. And there is a number of tumor types or tumor indications that could be of interest for Tumorad, and I've listed a few of them here. As you can see, they range from large indications like colorectal cancer until smaller indications like ovarian. And with ovarian, this is a very interesting indication that has come up several times during our assessment of potential positioning of Tumorad. There is a very high medical need in ovarian cancer, and also there is the opportunity to apply for orphan drug status in this indication. So we believe this is a very interesting indication for us to start positioning Tumorad in and to start to build kind of the clinical planning around. So a little bit more about this indication. The ovarian cancer, it's a lethal disease, unfortunately, affecting women, obviously. What it has is a demonstrated sensitivity to radiation therapy, which means that it is targetable by a radionuclide treatment like Tumorad. There is a positive EPR effect that has been clinically validated in this indication, which means that we actually do have the opportunity with our technology platform and with Tumorad to target these types of patients. The medical need, as I said, is huge. The 5-year survival rate is relatively low. They often metastasize, these types of tumors. Patients often present late, which provides a clinical challenge. But with the current treatment regimes that are based mainly on chemotherapy and 1 or 2 targeted agents, there is a remaining opportunity beyond those standard treatments to go in with additional treatment and potentially prolong the life of the patients. So what we are doing now to position Tumorad in this indication is doing a relatively ambitious preclinical program in an ovarian cancer model, where we are also testing combination treatments with Tumorad and standard chemotherapy and standard PARP inhibitors. So this program is aimed to support an orphan drug application with the FDA later this year. Looking into the market, and now I'm still staying with ovarian cancer. So this is the orphan indication, ovarian cancer, and looking at the market potential or the valuation of the project with current estimates that are rather conservative and with all the discount factors that applies to a preclinical project at this stage, we still end up with a project valuation that is significant in this orphan indication of ovarian cancer. And it rapidly increases, as we can see, over during the course of development, and at the end, the target, the total available market or the targetable market here is really significant. And remember that this is still one orphan indication, and Tumorad has the potential to be used in multiple indications as we go along. So what we see now, and I've shown this picture before, but there is really an increasing interest in the radionuclide therapy field. And we've seen this not only in the literature as shown here, but also in the deal flow recently. There has been large investments into this field, both on the private side, but also in mergers and acquisitions. We know that Novartis has been out there buying projects. We know that Astra has been doing investment in this field, and so on and so on. So this is now a field that is taken off. And we, as we all know, that ionizing radiation works for treating cancer. That has been known for many decades now. The trick is just getting it to the tumors in the body, and this is where we can make a difference with Tumorad. So if you compare Tumorad, lutetium to SN201, compared with peptide receptor radiotherapy, which is a more targeted or differently targeted type of radionuclide therapy, like Lutathera, for instance, you can see that with Tumorad, we are using physiological targeting, which is independent of receptor binding, which means that it's a more general mechanism than compared with antibody or peptide receptor targeted agents. We are using slow targeting with long circulation times compared to the more faster targeting peptide receptor radionuclide therapies. With slow excretion compared to fast excretion. And importantly, we expect Tumorad to be less sensitive to tolerance development. So that means that tumors will be likely to be less prone to regrowth or recurrence with Tumorad. And we can allow ourselves to go with a higher systemic dose. And I should also say that with Tumorad, this concept being more generalized than more biologically targeted agents also open up for combination, because it's long been known that radiotherapy can open tumors to other types of therapies. So that's why the combination studies that we are currently performing in the preclinical setting is so important. So we really believe that Tumorad is standing out if we compare with the pipeline agents or with agents that are already approved. And this is just a way to try to visualize that. We are using beta radiation. And beta radiation, as compared to alpha radiation, which is also used in radionuclide treatment, is better to treat bulky diseases. It has a better penetration potential, which means that we could potentially reduce the risk of recurrence with the tumors, so that will so to say kill the whole tumor. And then on the other axis, we have biological targeting versus physiological targeting, which I was talking about earlier. So biological targeting by means of antibodies or peptide receptor targeting is the general way that has been used by many of the pipeline agents and many of the approved agents so far. Whereas physiological targeting, and the drawback with that is, of course, that you can only treat one tumor indication at a time. So for instance, an antibody that is directed towards PSMA or other types of biological targets can only be used to treat that specific type of tumor. Whereas Tumorad can be more broadly applied where the EPR effect is documented, and that can be documented through imaging, for instance. Then we have other agents out there. Xofigo is perhaps the more classical example that are using alpha radiation. And alpha radiation has a much shorter span of penetration, which makes it limited in use to very specific types of tumors. So we really believe that Tumorad is standing out, and that there is a way to position Tumorad compared to existing agents and in combinations with agents in different types of solid tumors. So with that, we'll switch to our other project, Spargopix, which is now also switched from therapy to diagnostics. And as you know, Spargopix is our tumor-selective contrast agent for MRI. And I will hand over to Paul once more to go through Spargopix. So Paul, please.
Thank you. So you'll recognize this as a very similar slide to one that we showed slightly earlier, where we have the functional nanoparticle, but this time we use manganese for the precision imaging rather than for tumor a radioisotope. Next slide, please. So here, the clinical concept is very much that we have used manganese as a paramagnetic, which will provide excellent MRI contrast. That's already been proven. That we have, again, functional nanoparticles that have been optimized for the physiological targeting and retention within the tumors. So one of the issues currently with MRI imaging is that the contrast agents are injected very early in the process of the imaging, and they very quickly disappear from the body. With SN132D, Spargopix, we actually have something that can be slowly accumulating, giving the actual radiotherapy lab, the imaging lab, apologies, time to actually get good images. This is through that selective accumulation in the tumor and also metastases. And because of this retention, it gives us more time to actually get the images than other contrast agents that are out there, which allows for a much higher resolution of imaging over several hours. And this is important, and we'll see that in a moment. So next slide, please. So again, the differentiation points that we have with Spargopix are that we have selectivity for malignant lesions. So again, through this physiological targeting, we can get good precision MRI images. With no background enhancement occurring, we then reduce the amount of false positives that may be seen on the images, currently a big problem. Because the Spargopix stays within the tumors for a reasonable period of time, we actually can show that there is a reduced MRI scanning time because multiple images can be taken without the need to redose the patient with a contrast agent. We have flexible imaging, so there's the option for repeated scanning, again with high resolution, without that need for redosing because of the long effect. And we use manganese as our main metal, so therefore we do not have any gadolinium, which means that there's no retention of the metal, which has given some safety concerns in the past for gadolinium-based contrast agents. Next slide, please. So, as Matt's mentioned, we have released information about Spargopix in the past year. We had interim results from our first 12 breast cancer patients, which we recruited in 2 hospitals in Sweden, in Gothenburg and in Uppsala. It confirmed the previously presented results in the first 6 patients that we do see good accumulation in the breast cancer solid tumors. We've got a great safety profile in the higher dose level, so we don't see any concerns with dosing patients at that point. And importantly, we get clinically relevant contrast-enhanced images of the breast tumors, and we're seeing this in the pancreas and liver as well. So for that reason, we've actually expanded the inclusion for this study into patients with pancreatic cancer that have liver lesion involvement. And this is a significant unmet need that, because of the limitations of other contrast agents, it's very difficult to actually see smaller liver metastases for these patients. And this is somewhere that the clinicians, when we've spoken to them, see that Spargopix could really be of benefit. Next slide, please. So here we have some images, and hopefully you can see from the left-hand side, we have 2 breast images. The first one is pre-dosing with Spargopix, so you see a very dull sort of image of the tumor, making it difficult for a surgeon to actually assess the limits of the tumor. But on the right of that picture, you see quite a bright, enhanced tumor, which is where we have the Spargopix 2 hours after the actual infusion has completed of the Spargopix. So we're confident that we are seeing good images in solid tumors, and that this opens up at the use in other tumors. The image on the right is also of interest. It shows both the actual tumor, the malignant tumor, but also it can be seen by the reader areas of necrosis that are occurring within the breast tissue from that tumor. Having seen the tumors imaged in this way across the 12 patients, we're confident that we are seeing an EPR effect in the breast cancer tumors, which is one of the reasons that we believe that we will be able to use Spargopix to image many other types of solid tumors. Next slide, please. So as I mentioned, we've now opened up the study for pancreatic cancer patients. You can see on the image on the right that we've got a very nice imaging comes through after Spargopix. We've been told and understand that there is a major clinical need to identify metastases within the liver. The early detection of these smaller lesions and the follow-up of these precancerous lesions is also incredibly important for the survival time for these patients. And again, this is where the potential for this high-resolution imaging is really important. The ability to have the time within the MRI scanner to actually get images of this quality is incredibly important for these hard-to-see lesions. Currently, there is no available MRI contrast agent that is specifically indicated for pancreatic cancer. And with the expansion of Spargopix 01 protocol to this, we really hope that we'll be able to, in relatively few number of patients, prove the utility of Spargopix in this patient population as well. Next slide, please. Oh, back over to you, Mats, actually.
Thank you very much, Paul, for a comprehensive walkthrough of the current data with Spargopix. And as you all can see, these are very positive data. It's really encouraging for us, not only for Spargopix, but also for Tumorad, as it validates the whole platform per se. I should also say that with these results, we have actually reached what we aim to achieve with the current clinical trial in breast cancer. We have good and positive contrast and selective contrast consistently throughout the patients that has been included, and we have good safety with Spargopix in all the patients. So with that, we are very happy with the study so far, and what we've done now, and I will come back to that in a second as well, is to open up and use in an effective way the study to also look for broader signs of efficacy in these patients with pancreatic cancer. So what we've also said with Spargopix is that we aim to find a license partner for this project early on in clinical development. So we started that process last year, and we have had several discussions. We have evaluated the let's say players on the contrast agent market and all both existing and potential ones. We have done a lot of presentations to all of these, and we are now at the stage where we are into dialogue and discussions and around positioning and planning of the project. And that will hopefully lead later on to an agreement and partnership. So importantly, the feedback we've got so far, and this is two major points, I think. The first one is very positive, saying that what we've actually shown so far indicates that this contrast agent, that SN132D actually might be a breakthrough drug. And this is based on the fact that we have no background enhancement. It's very, very clean. We have contrast in the tumor and not in the background, and providing a very good image of the tumor itself. And the second point here is that if we can show that this is not visualizing benign tumors as well, so that we can make a true differentiation, that will also be part of a kind of a breakthrough perspective of SN132D. And to be honest, we've actually seen that as well, because we have had patients with liver cysts, for instance, and there is absolutely no contrast in those. So it looks as there is a good differentiation between malignant and benign lesions. One other piece of feedback that we have received is that many discussions has revolved around broader applicability of Spargopix. As you may remember, we entered into breast cancer because it's a suitable indication. There is a high clinical high clinical need, and MRI is already used as a standard technique in breast cancer. So there are good reasons to go into that, and now we have proven that it works. So the question is, well, what about other tumors? Will it work, as you say, in other solid tumors? We definitely believe so, and as I said, we see good correspondence between preclinical data and clinical data, but we'd also like to explore that in the patients. So that's why we are using the current study as a very effective way of including patients with pancreatic cancer to provide imaging also in another type of solid tumor, and potentially in patients who have liver metastasis as well. So that is ongoing, and we are also evaluating additional indications, and we have really interesting indications that there are certain types of non-cancerous indications, endometriosis for instance, that could be really where there is a high medical need and that could benefit a lot from use of SN132D. So that's also being evaluated right now. So to summarize all of this, we believe that we have proven that we have a solid platform to address major medical needs. We have a proprietary technology. We recently filed additional patent applications for Tumorad. We already have a very solid patent portfolio around both projects. That covers physiological targeting with our functional nanoparticles. We also think that the development has been de-risked very much by the results that we have seen with Spargopix in cancer patients. So it not only validates the platform, but it also de-risks development going forward, especially for Tumorad going into patients very soon. The product candidates we have are first in class, both for precision imaging and radionuclide therapy. And the mechanism we are using, the physiological accumulation of particles in tumors, is very suitable for combination therapies in different cancer indications. And we're now also optimizing the development pathway for Tumorad by aiming for orphan indication by means of ovarian cancer. So the activities that are now ongoing and is really following our plan and following on previous communication as well, is to prepare Tumorad for clinical trials and the aim to start, as Paul was saying, a clinical phase 1-2 trial by the end of this year. Also for orphan drug preparations, these preclinical studies are ongoing, and we aim to file the application during the second half of this year. On top of that, we are also intensified our discussions with specialist investors to position Tumorad and to prepare for future financing of the project as it will quite quickly come into clinical development stages that are more advanced. As I said, we are continuing to evaluate partnering opportunities for Spargopix, and we have that doing that, or we are doing that in ongoing dialogues, and also try to, in a very cost-effective way and as fast as possible, show broader applicability of Spargopix. So with that, I will come back to this image. I think we have shown that we have a technology based on a proprietary platform that is suitable both for precision diagnostics and therapy. We have the clinical evidence that the platform technology works. We can manufacture nanoparticles and we can make them land in tumors and do what we want them to do. Our pipeline is based on first-in-class products, both in diagnostics and therapy. And the market, as you've seen, is substantial, both in diagnostics, but particularly in therapy with billion-dollar potential. We did not touch very much on the team, but I think we have a very agile and experienced team, both in the management, in the employees, and in the board, so to execute on these plans. And the position we have a very strong ownership. We are listed at Nasdaq First North, and we're a stable and good company. So. With that, I thank you very much for the attention and open up for some questions.
Yes, thank you so much, Mats and Paul, for a very interesting presentation indeed. And as an analyst, you always get a little more exciting when you are talking about partnering and stuff like that. So I would like to start with a couple of questions about these, is it early stage partnering discussions, would you call it, that you have had so far for Spargopix, or?
Well, define early stage.
I mean, is it how much do they know about Spargopix before you initiate these discussions?
We've been quite open, and of course there's been different levels of discussion with different players, but I would say some of them has been rather advanced.
Okay, great. And what kind of reaction do you get when you show them the pictures and the data from the studies?
I think consistently everyone is quite impressed by the breast cancer images, and especially the fact that there is no background enhancement almost whatsoever, which is, as Paul was explaining, is really important to have these images. It is one of the limitations why MRI has not been more broadly used, and also it opens for more high resolution imaging and things like that, so you can get much more information out of the images.
Okay, great. And some of the comments you mentioned from the partners is that they would like to see perhaps more data from other indications. How do you interpret that kind of comment? Is it because they want to see a broader potential for Spargopix, or is it that they want to validate the data from a different tumor type?
I think it's both, actually. If you look, obviously, they would want to see that this works in different solid tumors, and that the concept with the EPR effect stands not only in breast cancer but also, for instance, in pancreatic cancer. And so there is a scientific rationale for that, which makes sense. But also, I think, looking historically, these companies are used to gadolinium contrast agents that can be widely applied from orthopedics to brain imaging to cancer imaging. Not that they are very good at cancer imaging, but they are used there. So I think that there is a, call it traditional view on how a contrast agent is going to be used, which we are now kind of changing a little bit with with Spargopix, as this will be a more specialized product. Having said that, there are specialized products on the market for liver imaging, for instance. So I think that this is kind of a way to gauge what is the maximum potential of the project, and I believe we have very good reasons to believe that it can be widely applied.
Yeah, sounds nice. I mean, if my impression is correct, this is a market where you don't have that many players, at least not larger companies, and given how they perhaps view, then they want to see more data and perhaps more potential. I mean, is it your impression that this is companies that are likely to strike early stage deals, or maybe you need to show perhaps a phase 2 studies?
I think there are opportunities for both, depending on what you agree upon, of course, and who is doing what and so on. So I think it's more about how do we position this product given all the other products out there. I mean, with Spargopix, we are, to my mind at least, we are collecting the best features of both PET imaging and MRI imaging into one, which means that this is a product that can, in some cases, perhaps replace PET, and in some cases replace MRI. Yeah. But you have to kind of show that, and you have to find the right target indications for that.
Perfect. And listen to your presentation, I get the impression that you're very confident in the breast cancer data so far. And maybe you could describe what would be the next step in breast cancer in developing. Are you have you finalized the phase 1 in your perspective?
I think with the phase, I would start by saying that I'm very confident right now. I've actually not been this confident ever in Spago. I've been here now for more than 7 years, and this is the first time I really feel that this technology platform works. So that's a really good feeling, of course. Coming back to breast cancer, we have shown what we wanted to show. In the study, we have shown good safety, we have shown good images in breast cancer. So now the question is, does it work also in pancreas cancer and perhaps other indications, and how can we position it going forward? So it's more about that, I would say.
Great. And what I mean in pancreatic cancer, that's very rare disease. How could such a development program, could that be a very fast way forward to market in that kind of indication?
Yeah, it could. I think what you would have to do next is perhaps include several indications in coming phase 2s and phase 3s, or even going directly into pivotal stage trial depending on the indication. You can of course either go narrow in a very specialized indication, but you can also try to go broader in several tumor types and compare with gadolinium agents, for instance, to show superiority or at least non-inferiority. So there are different ways, and those are typically the topics of discussions we will need to have or do have already with potential partners. Then there is, as I briefly mentioned also, the opportunity to perhaps go into some certain indications outside of the cancer area, which could be a separate kind of positioning or operational agreement or partnership agreement or whatever. So there are different opportunities here.
Yeah. Perfect. And then let's jump to Tumorad, which is a very exciting program indeed. And I mean, you're planning to start, is it to initiate the trial later this year, or Is it to have the protocol finalized? And
No, the aim is to start and to.
And do you see any kind of manufacturing or preclinical hurdles before?
It's always dangerous to answer these types of questions. Right now, it's going reasonably well. We have shown before that we can scale up manufacturing and that we can produce GMP, and that process is underway also for Tumorad. Then, of course, it's never a straight road, but for now we are quite hopeful, as Paul expressed, also to be able to start later this year.
Perfect. And one of the interesting things, from my perspective, about Tumorad is if I understand things correctly, that you. When you are entering like phase 2A, I guess you will be able to screen patients that you believe will respond to the treatment. If you could perhaps describe how that would work.
Yeah, so this type of I forgot the wording now, but the companion diagnostics strategy. I mean, the good thing with Tumorad is that it's based on a radionuclide, a radioisotope, which means that you can use that for imaging per se. So lutetium, for instance, can be used for SPECT imaging. And that is what we are going to test during the phase 1 part of the upcoming trial. So already at the first patients and the first doses, we are doing SPECT. And based on animal experiments, it seems to work reasonably well, which means that SPECT can be used to screen for patients later on. There are also other opportunities here which we are exploring right now. For instance, we could load the Tumorad particles with a different isotope that make it even more powerful for imaging, either for SPECT or for PET. So there's opportunity for both. Another option, of course, is potentially to use Spargopix as a proxy for selecting patients with good EPR effect in their tumors. But I should also say that what we've seen so far in the clinic is a remarkable consistency in accumulation of our particles in the tumors, which is really countering what has been described sometimes in the literature that states that the EPR effect is heterogeneous and so on. We haven't seen that. On the contrary, we've seen very consistent data between animal experiments and what we've seen now in the clinic so far.
Sounds interesting. And talking about animal experience, you are conducting, I guess, a couple of animal studies. When could we expect to get some more information of how these have progressed?
Yeah, we have quite an ambitious program right now going on in ovarian cancer models. As I said, including combination arms as well. So the current prognosis is that we will see results, well, sometime after summer from that program. It's quite a long lead times in these experiments, but somewhere around that.
I mean, cancer treatment is often combination treatment. Are you aiming to do some both monotherapy studies as well as combination studies before you really try to sell the compound to a partner? I guess it's always better to sell it direct, but I mean, to build value.
I think the best way to build value is to develop it as planned. We're going as quickly as we can towards pivotal stage. So that's why I'm saying that the orphan strategy here is I think it's the right way to go for us as a small company. In that way, we can build value in an indication that is not the biggest, but it's substantial, and the medical need is high. And based on that, I think we open up very much for discussions with whoever is interested in the project, taking it forward, either in single indications as monotherapy or bigger indication and combination therapies. There are multiple options here, I would say.
Okay, great. And as was mentioned during the presentation, there is quite a lot of interest in the radiopharmaceuticals. And I just wondered, do you see any real competitors to Tumorad in development that targets the EPR effect?
Not really. No, as I showed in this slide here as well. I think the concept of physiological targeting, and this is, I'd like to come back to one of the first images shown here, that Spago Nanomedical, we're really coming from the intersection between radiology, nanochemistry and nuclear medicine. And I think that is quite unique, to be honest. We are not doing drug delivery, we are not doing antibodies, we are doing something else that is unique and that can be combined, if you wish, with the other ones. But there are no immediate and direct competitors as we see it. There will be, of course, competitors based on other modalities, but yeah.
But do you believe that is because that the EPR effect has been viewed as a heterogenic, as you mentioned? Or is it...
I think it's time for nanomedicine 2.0, to be honest. The concept have been around now for, what is it, 30 years almost. But it's been based a lot on drug delivery. And we are working with functional nanoparticles, which is different. So I think what we can see in the nanomedicine field and what we try to be a part of is the shift of how do we use the EPR effect, which is obviously there, in the best way to treat patients or to diagnose patients. And I think what we have in our technology platform is a good start of that.
And that feels like a very good ending. Thank you so much for the presentation, Mats and Paul as well. Thank you all who has listened in.
Thank you very much.